This module is about the discovery of the tricarboxylic acid or Krebs cycle. By the 1930s, Hans Krebs knew that organic acids, when added to tissue homogenates in a test tube or in vitro, would stimulate oxygen consumption. The acids were themselves oxidized, and there was a pathway of oxidation in which the number of organic acids were converted one to another. None of these redox reactions directly involved oxidation of the acid by molecular oxygen. So let's take a look at this pathway. Here are the acids, and as they are oxidized, you saw the oxygen slowly disappearing, being consumed. The pathway begins with citric acid and ends with oxaloacetate, whose fate was not known at the time. The pathway is catalyzed by enzymes, obviously. Most, if not all of them, were not identified at the time, so we'll just call them enzymes 1, 2, and 3, and 4, and 5, and so on. Pyruvate, one of the end products of glycolysis, what was originally called the Embden-Meyerhof pathway, which had been discovered earlier, the fate of pyruvate was not known. So once pyruvate was produced by glycolysis, no one knew quite what happened to it in aerobic cells. Some organic acids that are not normally used by cells were also tested in the homogenates, probably just to see what would happen. One of them was malonate. This acid was not oxidized by the homogenates, that is, it didn't disappear. Not only that, it blocked oxygen consumption altogether, so malonic acid is in fact a respiratory poison. Krebs made use of that phenomenon in ultimately elucidating the cycle. So let's look at this slide from the beginning one more time. He showed, Krebs did, that by adding alpha-ketoglutarate or citric acid to these homogenates in the presence of malonate, in other words poisoned homogenates, succinate would accumulate. In other words, citric acid and alpha-ketoglutarate added would be oxidized, but succinic acid would accumulate. And so from this, Krebs concluded that malonic acid was actually the mechanism of its poison action was to block the enzyme number 3 here, called later succinic dehydrogenase, the enzyme that would oxidize succinic acid. So malonate is a respiratory poison by virtue of the fact that it blocks succinate dehydrogenase. Malonate is structurally similar to succinate, and so is a competitive inhibitor of the enzyme succinate dehydrogenase, and you can check that out in your textbook if you like. Krebs then found something remarkable. If he added fumarate or malate or oxaloacetate, now these are 3 of the acids in this pathway that are past succinic dehydrogenase, succinate would accumulate. Krebs drew the only possible conclusion, namely that the pathway of organic oxidations that he was looking at was not a linear pathway but was cyclic. And here we see the cyclic pathway. The fate of oxaloacetate then is partially resolved, at least it doesn't have that big question mark after it. Next, Krebs pondered what the cycle would be used for, and he suggested it was in fact a supercatalyst. That's a reasonable conclusion. Cycles generally are thought to be by definition a futile cycle, at least a biochemical cycle. But the natural thought process says, oh, this is a pointless thing to be doing. Of course, in living things, nothing is pointless. This has evolved for a purpose, and Krebs was wondering what purpose could it have. He thought back a little bit and realized that the end product of glycolysis was oxidized, but nobody knew what the reaction was. So he suggested the Krebs cycle, well, the TCA cycle to him, was a supercatalyst for pyruvate oxidation. Well, to prove this, Krebs did several experiments which meant to test the stoichiometry between each turn of the cycle and the number of pyruvates that would be oxidized. If in fact they were linked, then one turn of the cycle should oxidize 1 molecule of pyruvate. That's expressed here. In the presence of malonic acid, Krebs reasoned, tissue homogenates should oxidize 1 molecule or 1 mole of pyruvate for every mole of succinate that accumulated. Krebs experimentally confirmed this prediction, and therefore that the TCA or citric acid cycle was indeed a supercatalyst for the oxidation of pyruvic acid. In other words, he would do the experiment and he would show for every mole of pyruvate that he added to the homogenate, a mole of succinate would accumulate. It's in the presence of malonic acid, which would block the reaction from going past succinic acid. At some point later, Krebs's cyclic oxidation pathway was shown to occur in the mitochondrial matrix. So when he was doing his experiments, techniques of cell fractionation hadn't yet been developed, and so it was simply a homogenate of tissues that was doing the reaction, and he didn't really know what part of the cell was doing it. Later we come to know, of course, that the Krebs cycle occurs in the mitochondrion, which means then, of course, pyruvate has to get into the mitochondrion. The enzyme that would be predicted by Krebs' supercatalyst notion for oxidizing pyruvic acid was never found, despite a lot of searching. Instead, much later in the 1950s, these two reactions, both exergonic, were discovered in the mitochondria. They occur in the mitochondria, and the first one is the oxidation of pyruvic acid by the enzyme pyruvate dehydrogenase. As you can see, it's an exergonic reaction, and the electron carrier that grabs the electrons away from pyruvate is NAD+, and it becomes NADH. And the other thing to point out here is that in this process, the first of the carbon dioxide molecules that are to result from glucose oxidation or glucose respiration has now come off, and since there were 2 pyruvates per glucose, we would say that 2 of the carbon dioxide molecules have been accounted for the overall combustion of a molecule of glucose. The other reaction is a second one, taking one of the products, acetyl S-coenzyme A, one of the products of the first reaction, and linking it with oxaloacetate, OAA, to make the first component of the citric acid cycle, namely citric acid. It is in fact acetyl S-coenzyme A, then, that is oxidized by the supercatalyst Krebs cycle. And the enzyme that does it is citrate synthase, shown here. So no mystery anymore. There is an enzyme that links the oxidation of an organic acid to the Krebs cycle, but the organic acid that's actually being oxidized is not pyruvate, but acetyl coenzyme A, or acetate. A word about coenzyme A, which has a sulfhydryl group, so we often just call it coenzyme ASH or CoASH. Well, first of all, it's a B vitamin derivative. In its structure, it has an adenine and the SH or thiol group. The full structure is not shown here, but your textbook, I believe, has it. I might be mistaken. Check it out. In its interaction with pyruvate, pyruvate loses a carbon, remember, to become carbon dioxide, and the remaining 2 carbons of pyruvate are the acetate. And the acetate forms a thioester linkage with the coenzyme A. Coenzyme A is often referred to as activating the molecules to which it binds in this way. So acetic acid is essentially an activated acetate. Now, activated is a nice word because it implies activity or active. Thioester linkages, like the phosphoanhydride linkages that we saw earlier, are high energy bonds, high energy linkages that release a lot of free energy when hydrolyzed. So the thioester linkage in acetyl coenzyme A has now captured, at least for the moment, some of the free energy that was originally in the nutrient glucose way at the top of glycolysis. Let's take a look at the highlights of the Krebs cycle. Remember, this whole cycle, everything you see here happens twice per starting glucose molecule. It is a series of reactions of which 4 are redox reactions. And take a look. During that cycle, 2 more carbon dioxides come off. For 2 turns of the cycle, that's 4 of them. That represents the rest of the carbon dioxides that come off of respiration. There is, in step 5, a substrate level phosphorylation. In higher organisms, that results in the production not of ATP, but of GTP. In a subsequent reaction, the GTP is actually converted to ATP. That is, GTP transfers its phosphate to ADP to make ATP. So the net result of this substrate level phosphorylation is to produce another ATP, that is 1 per cycle, or 2 more ATPs per glucose, per 2 turns of the cycle. Bacteria actually make the ATP right away. It's only the higher organisms that have this intervening GTP production. There's our thioester linkage on the acetyl coenzyme A, and I want to point out that the free energy of hydrolysis of thioesters figures twice in the mitochondrion in the oxidation of acetyl coenzyme A, once outside the cycle itself, or actually at the beginning of the cycle, in which acetyl coenzyme A condenses with oxaloacetate, a 2-carbon and a 4-carbon compound come together to make the 6-carbon citric acid. That is an energy-requiring reaction fueled by the hydrolysis of the thioester linkage. And the second place is actually in step 5. The energy to form the GTP in higher organisms, or the ATP in bacteria, comes from the hydrolysis of a thioester linkage. I don't show that in step 5, but you'll see it later. So the net result of one turn of the Krebs cycle is to produce 3 NADHs, as it says here, a single molecule of GTP in higher organisms or ATP in bacteria, and 1 FADH2, and the release of 2 molecules of carbon dioxide. Remember, just multiply everything in that box by 2 to the number of these things per glucose that you started with. And that brings us to the end of this presentation.